really my pleasure to introduce Dr. Adriana Hung, who is a nephrologist and epidemiologist at Vanderbilt University Medical Center in Nashville. Dr. Hung obtained her medical degree from Universidad Central of Venezuela, her residency at Albert Einstein College of Medicine, and her nephrology fellowship at UCSF, where I learned she knew Rudy, and I assume Yoshio as well. She obtained her master's in public health at Vanderbilt. She has received many grants from a variety of different institutions, including the VA, NIH, National Kidney Foundation, and her research expertise is extraordinarily wide-ranging, and we look forward to hearing her talk today. So, Dr. Hum, welcome, and I'll turn things over to you.
Thank you so much, Matt, for the introduction. That was great. I've been VA-based most of my time. And just to give you a little bit of an overview of what my research focus really is understanding the role of genetics and environmental factors in CKD progression and related outcomes. And I also have been doing pharmacoepidemiology in the VA for more than a decade. So I'm actually formally trained in pharmacoepidemiology at the VA. And so I had the opportunity to work in the Million Veteran Program, and the presentation that I'm going to do today, it's all the investigations are done in the Million Veteran Program. And for the Million Veteran Program, I'm the chair of the PMP committee. But the topic that I'm going to discuss today is APOL1 mediated kidney disease and the progress that we have made toward precision medicine. I think APOL1 is probably one of the greatest examples, not just in nephrology, but in genomics, of going from discovery to implementation. I think that we are in wonderful times right now where there are at least 3 drugs that have come around to treat AMKD that are either in trial or in the pipelines. Okay, and so this is the outline of my talk. I'll briefly touch base on the epidemiology of APOL1 mediated kidney disease, because I'm sure that most of us do understand well AMKD. I'll spend most of my time talking about the kidney disease, APOL1 kidney disease spectrum, and the concept of a second hit hypothesis, and touch base in gene-environment interactions. I will review a study done by us in APOL1, COVID, and NKI interactions, and then cover gene-gene interactions, and that will include sickle cell trait and other rare variants in APOL1 gene that modify the nephrotoxicity of the variants we know, so of G1 and G2. Very briefly touch base about disease mechanisms for the renal risk variants, as they are related to the new therapeutics. And so, as we all know, APOL1 was discovered for the very first time over about 13 years ago in the context of HIV-associated nephropathy. As you know too, HIVAN is the most severe form of AMKD, with people going into end-stage renal disease in about 6 months when it first started. So back then, Jeffrey Cobb and Cheryl Winkler, using admixture mapping, were able to identify a region in chromosome 22 that was associated to HIVAN. And about 2 years later, Martin Pollack in Harvard, using fine mapping, was able to pinpoint that that was APOL1 and identify the risk variants, so G1 and G2. And that was very important, not because it provided a gene that was associated with collapsing FSGS and other idiopathic forms of glomerular diseases, but because it provided a genetic risk factor that allowed us to predict kidney disease progression in individuals of African ancestry. And it really was something that we could apply to all forms of non-diabetic kidney disease. This particular study here, and I think all of you have seen it, right? Bipartite, always a landmark study. The curve on the left side is from the African American study of kidney disease, and it's just showing what we heard, that there is a 90% increase in the risk of progression into a renal composite of a 50% decline or ESRD. The curve that I like to always highlight is the one on the right. So the chronic renal insufficiency cohort survival analysis, just because the European ancestry individuals are included in there. And you can still see that there is a significant excess risk in African ancestry individuals, even with low risk APOL1. So it's just telling us that there's still important risk factors here playing an important role. And yeah, we can think that there are social determinants of health, but I think there are other African ancestry specific risk variants that need to be identified, being one of those perhaps an already identified sickle cell trait, but there are others. I'm sure you also are all familiar with the functions of APOL1, which really are not fully understood. But what we know is that it plays a critical role in the innate immune response. This is a cartoon of the protein. The different domains of the protein are named after the strip-analytic activity. The most important domain is the SRA domain, because it's the one that interacts with the trypanosome. And as we know, the trypanosome, and it was really the trypanosome Rhodian CNC, developed resistance to the trypanolytic activity of APOL1, and we were able to overcome that resistance through G1 and G2. So people that had G1 and G2 were protected against trypanosomiasis, but unfortunately for those that had two risk alleles, they also had an increased risk of suffering kidney disease. And so G1, G2 really come from West Africa, where sleeping sickness was endemic, and it moved to North America, Central America, and other areas of the world through the slave trade. This map here was done by Girish Nakardi from BioMe in Mount Sinai, New York, and it's just showing the worldwide frequencies of APOL1 risk alleles using data from the PAGE consortium. And so it is a common genotype affecting about 12 to 14% of African Americans in the US. This is data from different cohorts, so ARIC, Dallas Hart, MESA, Regards, very consistent, is about, 12 to 14%. But I wanted to share the data from the Million Veteran Program. Actually, Million Veteran Program have now enrolled 940,000 individuals. But for those that have genetic data available, is about 123,000 African Americans, but that had genotypes in APOL1 was 121,942 individuals. And so you can clearly see here that the prevalence is about 12.9%. So I feel that this estimate is current and accurate. So I think that's about the percent of people with African ancestry that have the risk genotype. I also want to highlight that about 46% have one risk allele, and that is important because new phenotypes are recognized, mostly the effect on endothelial function. And in certain circumstances, there is an increased risk of kidney disease and acute kidney injury with one risk allele. And that's important because I think we nephrologists have been very, very, very centered in that you have to be in a recessive model, and that, basically that you have to have a recessive model, and that is a podocyte disease, right? But this new endothelial phenotype that I'll show you, it's a little bit different. And I just wanted to share, this is just like data from the MVP. Many times I get asked by reviewers, are you sure there is no Europeans or Asians that have a high-risk genotype? And the answer is absolutely no. This also provides the opportunity to see the different types of high-risk genotypes. So you can be a homozygous for G1, a homozygous for G2, or a compound heterozygous G1, G2. And I don't know that is relevant, but it also shows that G1 is far more common than G2. But the good news is that not everybody with a high-risk genotype will develop kidney disease. And that's why we all work with the concept of a second hit hypothesis. And so gene environment and gene-gene interactions. And so I think the most important environmental factor that we knew was viral infections. And so we talked about HIV or HIVAN, but we know that parvovirus V19. I at least have had 2 individuals that developed collapsing FSGS due to parvovirus V19 being a high-risk genotype for APOL1. But it can be with anything, CMV, can even be dengue. And so this particular figure here was put together by Juan Carlos Vélez in Nature Review. And I think is really good because it helps you to understand the pathophysiology of AMKD. So any disease condition that increases cytokine release, and particularly increases the production of interferon, will increase gene expression of APOL1 in the polycyte, which will then proliferate and de-differentiate and pretty much just crowd the space, the glomerular space. And that's what gives rise to what we know as a pseudocrest, right? And so this is just HIVAN. But something that is important here to also highlight in the electron microscopy is that if you see tubular reticular inclusion bodies, it's usually something that is meaningful for a high inflammatory response. So no matter what the process is, if you're seeing these, that's being driven by interferon and cytokines. And so any disease condition that's doing this can give you AMKD. And when COVID was in its peak, I think we all heard many case reports and case series reporting collapsing FSGS and different forms of glomerular diseases in patients with high-risk genotype. But what I'm going to share today is actually not collapsing FSGS, but when I talk about general AKI in hospitalized patients. And so this study here was done by Al-Ali, but was one of the reasons that I undertook the investigation that I'm going to share with you. Al-Ali study put 5,216 individuals that were hospitalized with COVID, and some of them suffer AKI. And he showed that being of Black race, increased the risk of acute kidney injury and the risk of dying. And the risk of dying was higher in individuals of African ancestry here. So we can see the interaction in this particular visual abstract, but there was an important interaction. And so based on those observations that there was an increased risk of AKI and an increased risk of dying in individuals of African ancestry, and knowing that COVID-19 was a very inflammatory condition, we hypothesized that APOL1 could be playing a role in the garden variety of AKIs that we see in the hospitalized patients. And so we did this study in the Milligram program. At the beginning, it took a while to really get the sample size required. So we started with 1,000. That was the number that we decided on. But this study actually has about 990 individuals. These were individuals that were admitted with COVID-19. And we really had data to really demonstrate if they had suffered AKI. very few exclusion criteria. We only excluded people that did not have data to evaluate formally for AKI or that had pre-existing ESRD. And so what we observed is that people that had the high-risk genotype had about a 51.2% incidence rate for AKI compared to 37.9% in individuals of low risk. And these increases in risk, as you can observe here, is all based on doubling the risk of severe AKI. So there was no difference in AKI stage 1, but stages 2 and 3 AKI. And so we did more formal modeling, and really I wanted to do a subgroup analysis in individuals without CKD, because we know that during with COVID, having CKD is just a strong risk factor for very poor outcomes. And so we were able to observe that even in individuals with normal kidney function, the risk of AKI was about double, right? When you had a high-risk genotype. The association was even stronger for severe stages of AKI, and we could see that association with death. And so from this very simple analysis, we learned we got a lot of knowledge from this study. I think one of the highlights is that it provided the opportunity for precision medicine. You may recall that people were talking about giving 3 antivirals in certain trials. And we do know that you cannot give interferon to people that have a high-risk APOL1 genotype in those circumstances. And I think I highlighted these to several people that were very focused on the trial. So that's something that was very important. The other thing that I think was a message is that maybe in this very particular group of individuals with African ancestry, using cytokine blockers may be even more efficacious than in others. So it highlighted that we should consider in this particular group that intervention even more. And hopefully, with now the development of APOL1 blockers, we could potentially use those in the context of AKI due to COVID. But I want to say that we now are working, trying to understand if these risks that we observe with COVID is present in other inflammatory conditions in individuals that have a high-risk genotype. So meaning an individual that has sepsis that is in the ICU and is very inflamed, is that individual at high risk of AKI regardless of COVID. And so that's what we're working on. I had worked with Kathleen Sastak. I did the translational part of one of her papers or projects where she demonstrated that high-risk genotype, and this is her work, is animal models, right? Also generate an endothelial phenotype that is mediated through the NLRP3 inflammasome. And I just did a simple FIWAS for her and analyzed the different codes that are related to sepsis or systemic inflammatory response, and did observe the association, which I think Orlando had shown before in regards. My analysis is using a recessive phenotype. I think Orlando did an additive model. So that's what I refer to. I think when there is a component of the endothelial phenotype, there can be an additive model playing a role. And yeah, here, this is the one from Orlando, right? So he's using an additive phenotype. And he did observe the association of APOL1, high-risk genotype, and sepsis. And we just did an analysis The paper is under review that confirms this association, actually in BioView. And it's also behaving as a linear phenotype. I want to talk a little bit about sickle cell and about sickle cell trait and not sickle cell disease. I think we all know that sickle cell disease is associated with a risk of kidney disease. The reason why I wanted to look at sickle cell trait again, well, one is because we have a really large sample size where we can do more granular analysis, but also because sickle cell mutation came from the same region from West Africa, and it travels to other areas of the world together in the same way that APOL1 high-risk genotype did through a slave trade. So one of the thoughts we had is that it wouldn't be uncommon to have an individual that have both a high-risk genotype and a sickle cell trait. And also we had observed already in our GWASs, this is a GWAS of African ancestry individual and Latinos, that sickle cell trait and APOL1 both are always coming as associated with kidney disease. This is how many sickle cell traits and disease there are in the Million Veteran Program. I was surprised. So you have 8,191 traits and 15 sickle cell disease. I'm surprised because individuals with sickle cell trait are not supposed to be in the military. They cannot die. They cannot ride on a submarine. They cannot be on an air crew. So it was surprising that there was 8,191 individuals. I tried to compare what we capture by genotype and what we capture by codes in clinical diagnosis. And I think that's where there is a big discrepancy. There is a lot of missing diagnosis for sickle cell trait. And this is a phenome-wide association study of sickle cell trait done. Oh, I want to highlight that for all the studies that I'm showing here, we did remove the 15 individuals with sickle cell disease. That although it's just 15, we didn't want them to be driving our findings. So this is just the FIWAS of sickle cell trait. By the way, if you do FIWASs, the Phi codes are wrong. And you will find sickle cell disease inside sickle cell trait group. So you gotta be careful when you use Phi codes. But it's associated with all forms of kidney disease, and probably that's not a surprise. I know that Bessie Young has done work on that area. But because we had the numbers, what we did is that we grouped the individuals in those that had two APOL1 risk variants, those that had one APOL1 risk variant, and those that had none. And then each of those groups was divided in those with and those without sickle cell trait. And we can see an effect in individuals with one risk variant. And we had, we did our sequential models adjusting, simple adjustment, and then fully adjusted. But you can see here the odds ratios too, and perhaps the odds ratio, the strongest association with kidney, well, I'm saying kidney disease, this is actually a time to event analysis. This is a composite of a 40% decline in GFR or ESRD. And if you pay attention here, the odds ratio, the highest are when you have APOL1 risk genotypes. But if you have the sickle cell trait, you can see that it is even higher. So the highest risk is when you have an APOL1 high risk genotype and sickle cell positive. So I think that's important. And this is the cumulative incidence curves. This is high risk APOL1. This is one risk allele for APOL1 and sickle cell trait. So it wiggles here in the middle. An interesting analysis. I wanted to learn if there was other biomarkers that we could use in clinical medicine to predict risk. Okay, so there's many individuals with sickle cell trait that come to clinic. Are they all at risk of suffering kidney disease? And so we use the urine. We took the specific gravity. We defined isostenuria, and we included also hypostenuria. And we took the early urines that were available for these individuals. And what you can observe here, which I think is just really interesting, is that individuals with sickle cell trait, regardless of APOL1, there is a clear association with isostenuria. And that's important. I think is highlighting that maybe subclinically, but there is ischemia happening in the vasa recta, in that kidney. And I think we need to do more studies understanding the meaning of specific gravity in sickle cell trait. I know we knew this, I know we learned it in medical school, but I think an epidemiological study to see this biomarker predicts risk, I think is important. I think we need just to work also in urinary biomarkers. We just don't have too many. I also wanted to highlight that when you see proteinuria, that's not sickle cell trait, that's just APOL1. When we did the same analysis for proteinuria, the association of heavy proteinuria is only present in high risk genotypes of APOL1, but not with sickle cell trait. So this is along with what we know. Yeah, AMKD is a proteinuric condition. I mentioned that there are other important gene-gene interactions. This is something that David Freeman and Martin Pollock have described as a haplotype background. So what it really means is that there's many other rare coding variants in the APOL1 gene. Okay, these are the most common of the rares, and they had postulated that they will modify the risk of nephrotoxicity associated with G1 and G2. So the nephrotoxicity will depend in what background they have. And so there was a time where people were doing many analyses in these other coding variants, and they were not actually seeing the risk because of what I just said. They were doing it independently of G1 and G2. But Herbert Annan and David Freeman, they use human embryonic cells, and they analyze what these rare variants did in the context of G1 and G2. And so what they really do is they annulate the risk. So this is for G1 and this is for G2. So this had been done all in vitro and in cell models. And so what we decided to do was to see what that will do in a large human population. So we did a study in MVP, but we also used BioView and all of us as replications. So we had a total of about 150,000 individuals. And what we studied was to see how the nephrotoxicity of APOL1 risk variants were modified in the presence or in the absence of the P-N264K. And we selected the P-N264K just because it was one of the easiest that we could really analyze. But we have analyzed now others. And we can see that what I'm going to share with you is unique to N264K. The primary outcome was CKD and ESKD. It's a cross-sectional design, and we did many models. We also complemented our study with functional genomics, just to make sure that our interpretation was correct. And so what you're seeing here is in individuals that have a high-risk genotype for APOL1, and they don't have N264K. They are at increased risk of all kidney outcomes, as you know, CKD, ESKD, heavy proteinuria nephrotic syndrome, and FSGS. But in the presence of N264K, that risk is minimized. Actually, I'll show you the results of the study itself so that you can observe that in an easier way. The groups that we had were individuals. Yes, we included individuals APOL1 with and without. So individuals with high risk with and without, and individuals with low risk with and without N264K. And this analysis done this way is really to test formally for an interaction. So if you have a high-risk genotype and you don't have N264K, your odds ratio for end-stage renal disease is 4. But if you have N264K, your odds ratio for that is 0.73. So you could say it's a little bit protected, maybe, I don't know, but you can say that it's the same as if you were a low-risk APOL1 genotype, regardless of N264K. So N264K, it won't modify the risk in people that have low-risk APOL1 genotypes. And the same is observed for CKD. And if you can, if the graph is easy to understand. And so these are the replications, and you all know Kasian. So Kasian did BioVU, Alex Big did all of us. The light orange is without N264K. Well, this is CKD, this is ESKD. The dark orange is with N264K. So MVP, BioVU, and all of us. This analysis is only done in individuals with high-risk genotype. No interactions are tested because the sample size wouldn't allow that. But in the meta-analysis, you can observe the same effect that is seen in the primary analysis in MVP. So it's protected. Cumulative incidence curve showing the same. High-risk genotype without N264K, high-risk genotype with N264K. So that's the purple curve. So drastically different. So we decided that we needed to do these, we needed functional assays. And we worked with Eric Green. He's in Mace Pharmaceutical. Eric Green is a cardiologist, and he was a fellow at Brigham. So I knew him from before. And what this is, and I'm going to try, I think these are podocytes and these are hex cells. Gray is G0. This is podocyte survival. And if you have G1 or G2, these are cell models, right? The podocyte survival goes down. In the presence of N264K, the survival of the podocyte is rescued. This is the mechanism by which the viability is brought down. So what G1 and G2 does is they open a pore or an ion channel on the cell membrane, and there is an influx of calcium that comes in. The cell swells up and suffers apoptosis. If you express N264K in that cell that has G2, that POR doesn't have it. Okay. And so what we so this company, the Maze Pharmaceutical, they have done now extensive work, and they have done work in G1 a lot, because in humans, G1s is the most common. And they just wanted to make sure that the effect was identical in G1 and G2. But the truth is that it should be, looking at the results in MVP, I think it provides really strong data that this intervention, or any intervention that is blocking the pore formation or the ion channel formation in high-risk genotypes, should have a tremendous effect in preventing the occurrence of AMKD. In any event, that's what this is, the compound, and I don't know all the details of their poster, but it's in the pipeline, is MC301. And so, oh, sorry. It has a recording. I wanted to, so there is, I'm gonna just go here to this particular slide, because I was just going to go over the mechanisms. So there are other mechanisms that have been proposed for APOL1 nephrotoxicity, but none of them are currently being targeted by any interventions. Okay, there was another drug that you may have heard from, that was from Ionis Pharmaceutical, that was an antisense nucleotide molecule. That one had side effects, injection site reactions, and they were not able to overcome the problem. So I don't think that's going to be developing into any trials or any further work. But in any event, APOL1 could open pores in the inner mitochondrial membrane and produce mitochondrial damage. It can also produce misfold in the ER, in the plasmid reticulum, and cause ER stress. And it can alter the structure of the cytoskeleton and produce podocyte dedifferentiation. But as I mentioned, I don't think right now anybody is using those mechanisms as targets. You probably saw that there in March 16 in the New England Journal of Medicine, Vertex Pharmaceutical made publicly and published their results of the phase 2 trial. Their drug is now called inaxaplin. It is a small molecule and an APOL1 inhibitor. This particular study is very small. It's only 16 individuals. It's a very short-term study, only 13 weeks. There were only 3 nephrotic individuals. The rest were subnephrotic range proteinuria. And it was an open label, so there is no control arm, okay? Despite that, the results are exciting. The PCR dropped by more than 50%. And with that observation, they did move to phase 3 trials. I don't know, are you guys doing Vertex? There you go. And so that this is their publication, but I think I just told you what it's about. Ogo Ebuna had told me that the Vertex drug has a similar effect to N2640K. Those are his words. I'm not 100% sure. But so in summary, I think it's a common condition, it's a common genotype, it's a common risk with a large effect, right? So I think it's important to genotype people actually, and to recognize those individuals that need interventions. not sure at this point which individuals are those, but I think anybody that has low GFR, has African ancestry, you may just want to genotype them. Interventions, maybe you want to tight control their blood pressure. When to initiate drug, that's something that we won't know. But I think that treating early is important because the drug can only prevent the development of AMKD. And so I think this is a very important observation, and I think we also need to learn implications for transplantation. And perhaps if a donor has a high-risk genotype, maybe he can feel more comfortable in donating if you know that there is a way that you can prevent the risk of nephropathy if any signs are detected. But if a donor has a high-risk genotype, I think it should be mandatory that they get monitored more frequently. I have had internal conflicts in letting some of my patients have their family member donate a kidney, knowing that they're APOL1 high risk. And so I think that we just have to always genotype, make sure that the person that decides to do that is very well informed of the risks. And now I think with the availability of these drugs, maybe, I'm not sure, this risk can be obliterated if that person develops kidney disease. So it may increase the pool too of donors. So that's all that I have. This is my lab. This is my programmer and my 4 biostatisticians. These are all mentees and these are collaborators. Internal, external, I have many, right, as we all do. So thank you so much. If you have any questions. And this is from Salvador Dali. I just went to Florida, to St. Petersburg last week and saw this in the museum. And so it was interesting. So this is the spiral of the DNA by Salvador Dali. Well, thank you. So I'll take questions. Any questions, please?
Thank you so much, Dr. Hung. Really wonderful presentation. Open up for questions. And certainly anyone locally there with you in the room, Dr. Hung, if they have questions, if you could just repeat it to everyone.
Great talk. I'll ask a question, Adriana. I want to ask a question about that great AKI work, and wonder if you can speculate on how the high-risk mutations might cause AKI. You spoke to podocyte mutations and endothelial phenotype, but we think of AKI as a tubular injury. So how do you think the high risk may lead to AKI?
That's right. So I've been thinking on the endothelial phenotype side.
And maybe repeat the question. I don't know
Okay. So Ian is asking, what will be the mechanism of AKI in COVID or in inflammatory conditions in high-risk genotypes? And given that we have the podocyte-centric view, and then now we're coming up with the endothelial phenotype. I personally think is through the endothelial phenotype, and it's just creating an ischemic AKI. But I think there is a lot to continue to investigate. But that's what I think. A hypertension, yeah, is also like endothelial.
In the Million Veteran Program for looking at genetics, have patients also been required or asked to list their race? You know, when we look at Caucasian, Black, and I'm wondering how many people of African ancestry would have been Caucasian or non-Black race?
Yeah, so that number, we know it, and it's a very interesting question. So when MVP started, we started working with self-declare at race and ethnicity using administrative databases. But maybe many of the VA researchers know that there is a significant proportion of missing, okay? And so we start wondering, well, why should we not use the ones that have missing race when we can estimate ancestry from the genotypes, right? And so that's how we started. And so they created something called HAIR. And so what they done, HAIR is harmonized ancestry and race and ethnicity. That's what HAIR stands for. And what they did is really that was created to provide information in missing data. So all those individuals that did not have a self-declared race or ethnicity, they took it from their genes, right? And there is a genetic definition of not race, but of ancestry, which is very different because we don't know really what race is, right? And so they use a very formal definition, which is based on your genes, and imputed that in there. But then they had to harmonize. So what they did is that anybody that had declared something that was not going along with their genes, they gave it the ancestry from their genes. So at the end of all the effort, I think they just gave everybody the ancestry from their genes. And now we're doing something called the GIA, which is like, it's the same thing. It's like, has a geographic component, but is also ancestry, completely ancestry based. That GIA is really being done to have more granularity in what we call Asians, right? So it's dividing the Asians in South Asians and East Asians or unmature. With that, the Hispanics will never be fixed. That's a very difficult race. So that has to be an ethnicity. Yeah. But European ancestry and African ancestry are fully ancestry based. Hispanic is self-declared. Asian is ancestry based, and now it has more granularity. And American Indian is self-declared. Mm-hmm. I hope I answered the question.
Well, part of that is, can you have African ancestry and be Caucasian?
Well, I don't know, but
Okay.
it is not uncommon that in the VA you declare a race that's conflicting.
Okay.
Mm-hmm. But to your question, the analysis is done on ancestry. So it's on your gene-based race. Mm-hmm.
Thank
you. Yeah.
Other questions from the group? Pavan?
That was a great talk. I just had another question about the AKI piece. With APOL1 being such a strong risk factor for CKD and CKD being a risk factor for AKI, how do you tease out, is it APOL1 going through CKD and mediating this risk, or is it independent?
Hi, Pavan. Thank you for the question. Yeah, so very good question. The only way that I knew to address that was doing the analysis in people with normal kidney functions to begin with. And that's what we did, because I think it's impossible to, tease that out once you have CKD.
Could still be subclinical CKD, perhaps.
Oh yeah, it
Right.
could be still subclinical CKD. That's right. So I guess that the only way to really answer that question is to make sure that the analysis is done in people with documented normal GFR and normal urine. Mm-hmm.
Thank you.
You're welcome. But yeah, we just did an analysis on sepsis. Again, that I was similar to what Orlando had done, but in a larger scale.
Yeah, the sepsis work is really exciting because APOL1 is expressed on non-kidney cells too. And so it might have other phenotypes other than acute kidney injury and sepsis, other endothelial-based phenotypes. So that's great work.
So, Adriana,
Mm-hmm.
in terms of kind of the gene environment interaction, do you see any differences in kind of the sort of social determinants or social risks in people with like high risk variants with and without kidney disease, or even with people between people with single variant, two variants?
That's a really good
And
question.
Adriana, if you could just repeat the question for the group.
Yoshio is wondering on the gene environmental interaction part, gene environment, and he is talking about social determinants of health. And I think it's a really important question. So in my renewal, I thought about that, and I went into nutrition as one environmental factor. We know that unfortunately your eating habits are very different, right? In people that have low SES. And so that's one factor. But there's maybe many other factors. Can be environmental, environmental exposures, military exposures can be one, for example, right? That could be looked more carefully. But people have done many things, Yoshio, have done air pollution in APOL1, right? That was Biomi, Gideon Nakarshi. And they've looked at other risk factors that are completely environmental. I personally, I'm going to go for the diet right now. I think is a big one, an obvious one, right? That people don't eat right in low SES. And so just to tell you, my thought was everybody's talking about hypertension and high risk genotype, right? Well, I think that cell sensitivity needs to be looked carefully. And in those low ACS, that super salt consumption may have something to do. So that's where my hypothesis kind of went. Very important, but a very difficult topic.
That's another question too. So the N264K mutation is very interesting, very exciting. It sounds like the discovery of that supports the mechanism of the Vertex drug. Does it also open the door for additional therapeutics, or other therapeutics through that pathway?
Yeah, so all the pharmaceutical companies keep everything very secretly. So the MC301, I don't think is the same. It's not similar to the inaxaplin. They're completely different, but they are doing the same thing, right? So it's like having it's like having different ACE inhibitors, I would say, but
it's the same mechanism.
It's the same mechanism, just a different drug. Yeah, I mean they're doing a patent on their compound, and so there will be. I mean, they act similarly, but I don't think that any other mechanism has been pursued. Only that drug that I mentioned from Ionis Pharmaceutical that was basically shutting down the production of APOL1. So it was inhibiting the biogenesis of APOL1. I kind of didn't like the wide consequences of the drug, but yeah,
along those lines, do we know the prevalence of
it's low. Yeah. Yeah. So yeah, they're asking. Sorry, their name. Sarah. Sarah is asking, what is the prevalence of N264K? And it's really low. It's like 2%. So.
And that's people with African ancestry, or
that's right. So that's why other studies probably didn't see it, because you really need a really large sample size to see the effect. And people were thinking about these variants because they're all coding, but in order to see what they did, you had to have a really large sample size. But the other thing is that you had to explore them in the context of high risk, because they don't do nothing if you're not high risk.
All right, well, thank you so much, Dr. Hung, for this presentation and discussion. Really appreciate it.